Hello biologists! In today's daily revision task, you are asked to produce a concept map on enzymes and the role of enzymes in catalyzing reactions that affect metabolic or metabolism at a cellular or at a whole organism level. Hello and welcome to this A-level biology revision video on enzyme action. Right, some key things to remember about enzymes are that they're biological catalysts. Many questions in your A-level biology paper will talk about enzymes, but in all of them you will need to make reference to some of the key points about how they work and the general action of enzymes. Enzymes are biological catalysts. They increase the rate of metabolic reactions. Almost all biological reactions involve enzymes. All enzymes are globular proteins with specific tertiary shapes. They are usually specific to only one reaction. You may need to talk about how the shape of the enzyme builds up. Remember that the primary structure of a protein is made by a polypeptide chain. Its secondary structure is held either in an alpha helix or a beta plated sheet by hydrogen bonds, and in its tertiary structure it's folded up on itself and held together by disulfide bridges, and its quaternary structure, which is where it forms a globular protein, is made up by more than one tertiary structure combined together. The part of the enzyme that acts as a catalyst is called the active site. The rest of the enzyme is much larger and is involved in maintaining the shape of the enzyme and the active site. When a reaction involving an enzyme occurs, a substrate is turned into a product. The substrate can be one or more molecules. The active site of an enzyme is complementary to the substrate it catalyzes. So the substrate fits into the active site exactly. It's important to remember that not only is it the right shape, but there might be hydrophilic or hydrophobic bonding occurring within the active site. There might be van der Waals forces acting, or there might be charged forces almost acting as ionic forces which hold the substrate in place. Some examples of enzymes that you may come across are lactase, that break down lactose into glucose and galactose; catalase, that breaks down hydrogen peroxide into water and oxygen; glycogen synthase, that catalyzes the formation of glycosidic bonds between glucose molecules; and ATP synthase, which produces ATP from inorganic phosphate and ADP. Since enzymes are proteins which are affected by their environment, organisms that live in varying conditions have adapted by producing enzymes that are more suitable to their environment. Endotherms, which are animals that maintain their temperature, keep the temperature of their enzymes within their bodies constant to ensure an optimum rate of reaction. It's that phrase, optimum rate, simply means the best rate of reaction that can occur. It's important to make reference to it. Enzymes are useful for a wide variety of purposes, such as digestion and many others. The action of an enzyme may be intracellular, so the enzymes are attached to the cell membrane, or in the cytoplasm and reactions occur inside the cell, or extracellular, so the enzymes work outside the cells and their products may be absorbed into the cell. Enzymes are also used in protection against pathogens. They can be used to destroy invading microorganisms. For example, phagocytes engulf pathogens, and during endocytosis, the vesicle then fuses with lysosomes that contain digestase enzymes that help break down the pathogen. There are a wide variety of things that enzymes work for. This graph shows how enzyme action lowers the activation energy of a reaction. Without enzymes present, the activation energy within the body would often be too high for most reactions to happen very quickly, so products wouldn't form fast enough and the cells wouldn't function. So most cells or most reactions in a cell require very high temperatures to get going, which would destroy the cell. Enzymes work by lowering the activation energy of a reaction. The activation energy of a reaction is lowered by putting stresses on the bonds within a molecule or holding molecules close together. This increases the likelihood of reaction and so lowers the energy required to begin it. The lock and key hypothesis is a model of how enzymes catalyze substrate reactions. It states that the shape of an active site of enzymes are exactly complementary to the shape of a substrate. Right, below is a diagram showing the lock and key mechanism. You need to make sure that you are familiar with this diagram and can explain all of the processes that are shown by it. When a substrate molecule collides with an enzyme whose active site is complementary, the substrate will fit into the active site and an enzyme-substrate complex will form. The enzyme will catalyze the reaction and the products together with the enzymes will form an enzyme-product complex. According to this model, it is possible for an enzyme to catalyze a reverse reaction. A more recent model, which is backed up by evidence and is now widely accepted as describing the way enzymes work, is the induced fit hypothesis. It states that the shape of an active site are not exactly complementary, but change shape in the presence of a specific substrate to become complementary. When a substrate molecule collides with an enzyme, if its composition is specifically correct, the shape of the enzyme's active site will change so that the substrate fits into an enzyme-substrate complex. The reaction can then be catalyzed and an enzyme product complex forms. Like I said, this is a more recent idea, but is now widely accepted as it doesn't allow for an enzyme converting products back into reactants, which would be counterproductive within the cell. Make sure that after you've watched this video, have a go at some exam questions that talk about enzymes. Make sure that you go on and watch the other videos that's talking about enzyme action and the uses of enzymes that I will be posting. Right, good luck. Hopefully this video should have helped you complete your daily revision task. Please subscribe so you can keep up to date with other videos to help with your daily revision tasks. You can follow me on Facebook or follow me on Twitter to help you keep up to date.